Right, in this pre-lecture, we're going to talk very briefly about waterborne pathogens. So we're going to focus on some of the specific aspects of why we care about the pathogenic species when it comes to drinking water, and make sure that you really understand why that's going to matter going forward when you choose how to disinfect or choose how to treat. So the first thing that I want to make sure that we understand is how we classify waterborne pathogens. So we can classify based on the type of organism, so whether it's bacteria, protozoa, all the things that we've been talking about in class, or we can classify it based on its relationship to water and or feces. So that's called an environmental classification or a prevention classification. So many times, instead of saying it's a bacteria or protozoa, we're worried about whether it's waterborne, foodborne, associated with feces, associated with drinking water, because water, wastewater, feces, they're all different vectors to basically catch diseases from these pathogens. So that's more of an environmental classification, and that's common once you're starting to get into the industry a little bit more. So this is the environmental classification of a lot of our water-related infections based on how we want to prevent them. So there's many different transmission routes that we can have. We can have what are called waterborne, water-washed, water-based, or water-related to insect vectors. And this is important because they're all related to water, but how the pathogen actually gets into your system and makes you sick is actually very different. So for example, when we talk about waterborne, a person or animal is going to ingest the water. So the disease group is what we call a fecal-oral route. So it comes from fecal contamination and it's consumed orally. So preventing this type of disease is just increasing the water quality, make sure that it's really, really good, because if we're going to ingest it through this fecal-oral route, we want to be very, very careful. So some examples here could be cholera, typhoid, cryptosporidium. A lot of diarrheal diseases are waterborne. Then we can also have what's called water washed. So these are favored when we have inadequate water for hygiene. So we don't have enough water to wash our hands or make sure that we're doing what we need to do. And so this can actually be skin and eye infections. So having more water quality and better hygiene is actually a huge thing with relationship to making sure we don't have transmission of water-washed diseases. So these are different from waterborne diseases, and these can be things like scabies, trachoma. Water-based diseases are basically pathogens that spend part of the life cycle in the water. So they have an aquatic intermediate host. So it's a water-based disease, and basically we just want to make sure that we're not having contact with water or decreased contamination of water. We don't want things like worms or schistosomiasis. Oh my goodness, that's a big one. But that one can penetrate skin. So these could be more like dermal if we're like in a recreational water and we're worried about that. We can also have water-related insect vectors. So if we have insects breeding or biting in or near water, like mosquitoes, right? That's basically the insect vector that we have. So if we manage our surface waters, we decrease breeding sites, kind of like what we talked about when we were talking about Disneyland and decreasing the ability for mosquitoes to breed. That's related to a water-related insect vector pathogen. And things like malaria, dengue fever, yellow fever are all related to insect-borne diseases. So just the takeaway here is that it's not so simple in terms of saying it's a waterborne disease, because there are many ways that water comes into contact with us as humans, so we need to be aware of what the transmission route is. So we can also classify based on groups. We can say whether it's a virus, a bacteria, a protozoa. We also have another classification called helminths. These are worms. Yes, we're going to talk about worms in this class. And basically the reason we classify it by groups is because of all those structures that we talked about at the very beginning of class. If we understand their structure, we know how to disinfect these guys. So if we know it's a protozoa, we know that's going to have different structures than bacteria, and so we can design treatment for that. So that's going to be really important. And the other thing here when we think about treatment or inactivation is we want to think about whether it's removal, like filtration, depending on size and things like that, or inactivation, looking at removing or inactivating DNA, like we talked about with UV disinfection, those kind of things. The other thing that it's useful for is when we develop detection methods or life cycle, or understand their life cycles. So when we detect viruses, it's very different from how we detect bacteria and very different from how we detect protozoa. So if you remember way back to when we were talking about water sampling, we sample for protozoa and bacteria differently. So if we're trying to detect a very specific organism, we need to know what we're doing, basically. So the other thing that I want to make sure that we talk about is sizes. Sizes are important when it comes to these organisms. So when we talk about viruses, they're on the order of magnitude of 0.1 micrometers. Bacteria are on the order of magnitude of 1. Protozoan and cysts are on the order of 10 micrometers, and helminth eggs, so worm eggs, are on the scale range of about 100 micrometers. So size matters, right? Because if we use size exclusion for filtration, we need to know how big those pore sizes need to be in order to remove the things that we want to remove. So these sizes are going to be really, really important. And then a little review here, because it's been a little bit since we've talked about this, but remember that your bacteria, your archaea, your eukarya are very different. And so if we're thinking about how we want to remove these pathogens, right? Pathogens are it's just a word that means it causes disease, right? So you can have bacterial pathogens, protozoan pathogens, fungal pathogens, right? So just remember that they are in very different groups of the tree of life, so that they have different features and different things that we want to be aware of. So just a little review here. So we're going to talk about some common characteristics of some of the things that we might be worried about if we were trying to remove these pathogens from the systems. And we really haven't talked about viruses so much, but viruses are just DNA and RNA. They're not considered living still, and they are what are called obligate intracellular parasites. They cannot exist without another cell. So they basically, what a virus will do is it hijacks another cell's systems and uses another cell's systems to replicate itself. So they don't actually have metabolisms, which is one of the reasons we haven't been talking about them that much. So it's very hard to disrupt any cellular processes because they aren't cells. So that's very difficult. They're not sensitive to typical antibiotics, although there are some antiviral medications. So that's something that's really interesting if you think back to COVID, right? We couldn't use our traditional methods of disinfection for viruses except for maybe filtration because they don't have the cell components, right? So we couldn't use chlorine disinfection because it's just DNA in kind of a capsid when it comes to a virus. So that's important, right? And viruses also adapt to different types of hosts. So enteric viruses tend to be very specific to the type of host. So it could be a human virus, a cow virus, right? Those kind of things. They can jump species, and that's what we care about when we hear about things like the Ebola virus or viruses like the swine flu and things like that that are viral related. They can jump species, and that's a very big concern. We do also have what are called bacteriophages, which are viruses that infect bacteria alone. Bacteriophages are not going to infect us, so we're actually not worried about those as humans. One of the things that we care about with viruses that's so important to us is that they have relatively low infectious doses, although sometimes it needs to be high. So this was one of our big concerns when we had COVID, is you need a very, very low dose of that virus to get sick. So as a result, we wanted to be very, very careful with preventative measures to make sure people weren't getting sick, because a low infectious dose is something common to viruses. When we look at viruses, just because we haven't talked about them before, generally they have what's called a protein capsid that surrounds some kind of a nucleic acid. And so it could be RNA, DNA, there's many different funky forms of nucleic acids that are present in viruses, but they all have the backbone of DNA or RNA. And that protein capsid is really a protective measure. It's defined according to structure, so we might say that it's a helical structure or something like that. There's no envelope associated with virus, no lipid membrane, so they persist in the environment quite a while. And we can also have what are called enveloped viruses, and they have the ability to play a role in attachment or release of other viral particles, and they're more sensitive to heat and desiccation. So just something interesting about those ones. So some key points about enteric viruses and why we care about this. The fact that they don't have a lipid membrane or they don't have lipid membrane or other cell structures is hard for inactivation or removal, because a lot of our traditional methods are all about breaking up those membranes. They do have capsids that are porous, so sometimes we can inactivate by trying to push something inside those capsids, but it's a little bit difficult. The capsid surface characteristics tend to be very specific, and the virus size tends to be very specific, and so that affects removal. So it's very hard to find a generalized method for removal of viruses. So that type of genomic material and the type of proteins that they have can affect susceptibility, basically, to inactivation. So one thing about viruses is that they can't grow without cells, so they need a host cell, and that's really important. So they don't just grow in the natural environment, they must have a host cell. That's what it means to be an obligate parasite. And basically, we need to, if we're going to try and detect viruses, we need to be able to detect the host cells of the viruses. So that's really important. So there's a lot of methods related to bacteria, to mammalian cells, to try and be able to find these viruses, and many of them are not culturable in a lab. So that's part of the reason that when we see a lot of our drinking water disinfection, we're seeing a lot of methods related to filtration, reverse osmosis, very, very fine membranes based on size, because it's very difficult to inactivate using UV or chlorine disinfection. So let's talk a little bit about bacteria, come back to why we would also care about bacteria. So just a little brief review on some of the structures and things that are important as pathogens. Remember that they're prokaryotic, they replicate by binary fission, and we can have species or strains that often infect humans and animals. This is kind of why we care about indicator organisms, right? And one thing about bacteria that's important for us to know as a pathogen-related aspect is that the infectious dose is actually usually very high. So we actually need a lot of bacteria in order to infect you and get you sick. So you might, for example, ingest one E. coli cell and it might not make you sick. But if you ingest a lot of E. coli cells that are present in water, that is much more likely to get you sick from these type of bacteria. So that's something that I wanted to make sure that we talked about with bacteria before we move on. So if we think about their structures and we think about how we're going to remove them or disinfect them, they're generally easy actually to inactivate by disinfectants. That's where a lot of our technologies have been developed. But the hard part is when they're in biofilms. And again, that's a recent kind of upcoming area of study that we're caring about. If they're in biofilms, floc particles inside protozoa, in spores, then it just makes it more difficult to inactivate. So in general, if it's the cell by themselves in an aquatic environment, chlorine, UV, they work just great. But we can have a lot of complex surface characteristics. We can have them in biofilms, and that makes them harder to inactivate. Excuse me. And as we've talked about in the past, normally they have a net negative charge on the outside. Remember when we talked about cation exchange capacity? They have that net negative charge. So they are actually something that can be coagulated and flocculated out of a system. They have high potential to grow in environments, so we know they're going to grow in environments. So that's important to us. They don't need the host cell like a virus does. And then if we're going to try and detect bacteria, we need to have the right culturable conditions, right? We've talked about that quite a bit. What type of agar you have, what type of filter you use. Remember that we really can't culture a lot of bacteria. So pathogenic bacteria actually tend to be very difficult to culture in a lab, unfortunately. And we can have what's called the status called viable but non-culturable. And that's a type of organism where they're still living and they're in the sample, but we can't actually culture them. And one example of this is Vibrio cholera, which is cholera. So we can't actually culture cholera in the lab, but we know that it's living in a sample. Last little characteristics for us to go over when we're thinking about pathogens is protozoa. Many times these types of protozoa are parasitic. So remember that they're eukaryotic, they're single-celled, they have complex structures, they can be free-living or parasitic. And sometimes when we group them by mode of kind of transportation or how they obtain food. So we can have flagellates, which have flagella, we can have amoeba, we can have sporozoans, which produce spores, or ciliates. And the thing about protozoa that's relevant to us for pathogens is that they have very complex life cycles. So they produce spores or they produce cysts, and it's actually the cysts associated with protozoa that are infectious, not the cell themselves. So that's going to be really important, and we'll talk about that more in class. Protozoa can infect both animals and humans, so Giardia is a good example of that. And the thing that we care about from an infectious dose perspective with protozoa is that the infectious dose is usually low. You only need to get a couple protozoan cyst cells inside of your body for you to get sick from these guys. So that's been really, really important in probably the last, I would say, 20 to 30 years in terms of disinfection. So that's important here. Then the last thing that I just want to mention is kind of these cysts. So protozoan cysts, or what we call oocysts, are smaller and they're very hardy. They tend to evolve to survive in the environment and persist in the environment, kind of like endospores for bacteria. They have very thick cell walls, so they are actually highly, highly resistant to chlorine disinfection. They have a net negative charge, but they are very infective once you've excreted them from your body. So once protozoan cysts are in feces, they are very, very infectious. These cells can't multiply by themselves in the environment, but they are kind of like viruses in that they can be parasitic once they get inside of you and they multiply inside of a host cell. So that's going to be important to us as we get into class and talk a little bit more about pathogens, because the life cycles are so complex with these types of cells. So if you take away nothing from this lecture, take away the idea of infectious dose and how infectious dose is different. Remember that size is going to matter in terms of the type of pathogens that we have. And remember that the type of organism or the type of vector that the organism is entering your system and causing disease is going to be different. That's going to wrap us up for pathogens, and we'll talk more about this in class and kind of bring this all together with a lot of our indicator organisms and drinking water concepts.